So this is one of the Royal Society of Biology's Policy Late events, and the series is supported by the Policy Lates working group organisations, and they are the Biochemical Society, the British Pharmacological Society, the Society for Applied Microbiology, the Society for Experimental Biology, and the Physiological Society. The Policy Late events are supposed to engender informal debate on a topic of interest to the biosciences currently. And the event is specifically here to enable knowledge exchange and interdisciplinary and intersectoral interaction on important bioscience policy topics. Of course, we have the current pandemic situation, and so this series is normally face-to-face. Of course, we're doing this online. This is the 6th online series in the event and the 2nd of 2021. And any feedback from the audience is welcome after this event. Recordings of previous Policy Late events can be viewed on the RSB YouTube channel. And topics include educational transition, climate crisis, healthy ageing, and antimicrobial resistance. For the audience, can you please send all questions for the panel through the question and answer box? These will then be put to the panel after the final speaker has finished. I have to say that there may be some questions that we can't answer simply due to time constraints, but we'll try to get through as many as we possibly can. I'd like to welcome our speakers today, who are Teresa Lambe from the University of Oxford, MacLeod from the University of Glasgow, and Ankur Mutreja from the University of Cambridge. And I'd also like to thank the organisers of this event, Beth Morgan, Laura Marshall and Karen Patel. And I'd like to thank you, the audience, for attending. I just want to give you a very brief opening few slides to talk a little bit about the history of vaccinology. So first of all, before I start, I have a conflict of interest to declare. I actually have a research project which is part funded by AstraZeneca, the maker obviously of the AstraZeneca COVID vaccine, but the project that I'm working on is on the molecular virology of rhinovirus infection, and that's the virus that causes the common cold. So traditionally we have a number of different vaccine types, and I'm sure Megan will go through this in more detail, but just to let you know, we traditionally over the last few decades and in fact centuries, we've been using inactivated vaccines, live attenuated vaccines and subunit vaccines, and there's some examples on the screen there. Sorry. However, the new vaccines that we are currently rolling out against COVID-19 are two different types. They're viral vector vaccine, for example the AstraZeneca COVID-19 vaccine, and nucleic acid vaccines, for example the Pfizer-BioNTech or Moderna vaccines that are being delivered in the UK. So what about these new vaccines? And something that struck me when I was thinking about this webinar was that in the media there's a lot of interest in the fact that these vaccines seem to have come from nowhere. There's vaccine hesitancy because people are a little bit wary of them, but in fact have they come from nowhere? So let's look at adenovirus as a vaccine vector. Adenovirus was in fact discovered in 1953. And over the last decades, it's been thoroughly characterized at a molecular level. As virologists, we know all we need to know about adenovirus, well, nearly. And there have been decades of development for vaccine delivery using adenovirus as a vector for cancer treatment and also for treatment against infectious agents. And adenovirus is particularly good as a vaccine vector because it induces good innate and adaptive immune responses. And I was really struck reading the Financial Times Weekend magazine, not this weekend, but the weekend before, of this cover article, The Messenger, dealing with the development of messenger RNA vaccines. And the cover title says, How Messenger RNA Went from Mystery Molecule to Vaccine Game Changer. Now, I'm not just a virologist, I'm also an expert on RNA biochemistry, and this really amused me because in fact, people have been working on messenger RNA for 6 decades, and there must be tens of thousands of people in the world who would argue that messenger RNA is a mystery molecule. In fact, there have been 3 decades of very intensive research on in vivo RNA delivery as a vaccine. And we know that messenger RNA shows great promise in the field of vaccines against cancer. And of course, we now know that messenger RNA as a vaccine has been delivered against COVID-19. And there has been strong proof of its efficacy in raising an immune response against infectious agents. And the data are looking good for its efficacy against COVID-19. But there are pros and cons of the two different types of new vaccines. The messenger RNA vaccines are great because they're non-infectious. They don't induce anti-vector immunity because there's no virus going into the body. They can be very rapidly designed and redesigned, and there's inexpensive scalable manufacture. However, the messenger RNA vaccines require frozen storage. Adenovirus vaccines, these are live viruses delivered into the body. They have a very broad tropism, which means that they infect a very wide range of cells in the body. They're very durable in the body, and they can be very easily prepared. They're good because they're relatively thermostable, not like the messenger RNA vaccines. And they do, as I said earlier, induce extremely good immune responses. But pre-existing immunity to this group of viruses may induce efficacy. So that's just a little background to today's topics. We're going to hear first of all from Associate Professor Teresa Lambe, who's going to talk about translational research successes and lessons learned from COVID-19 vaccine developments. We're then going to move on to Dr. Megan MacLeod, who's going to talk about the immunology of vaccination. And finally, we're going to hear from Dr. Ankur Mutreja, who's going to talk about the implications of COVID vaccine development, innovation in potential for developing new vaccines and other aspects of the biosciences.